Hello, everyone. Welcome to today's live broadcast, Clinical Next Generation Sequencing for Precision Cancer Therapeutics. I am Brenda Kelly Kim of LabRoots, and I'll be your moderator for today's event. LabRoots is the leading scientific social networking site, and we are proud to bring you this interactive web seminar. For more information, please visit us at www.labroots.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit your questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to get to as many of your questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation properly, please let us know by clicking on the support button at the top right of your presentation window, or submit your problem through the green Q&A button lower left. This presentation has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. I would now like to introduce today's speaker, Dr. Christina Lockwood. Dr. Lockwood joined the Department of Laboratory Medicine as an Associate Director of the Genetics and Solid Tumor Diagnostics Laboratory at the University of Washington Medical Center in 2014. She is board certified by the American Board of Clinical Chemistry and the American Board of Medical Genetics and Genomics. After obtaining her PhD at the University of Wisconsin, Dr. Lockwood completed postdoctoral fellowship training in clinical chemistry and clinical molecular genetics at the Washington University School of Medicine in St. Louis. Her interests include the development and application of molecular methods for the detection of genomic alterations in genetic disease, hematologic malignancy, and solid tumors. Dr. Lockwood has also been active in the design and implementation of educational resources related to training in genomic medicine. I will now turn it over to Dr. Lockwood for her presentation.
Thanks very much, Brenda, for that kind introduction. It's my pleasure to join with you this morning as we are going to be talking about clinical next-generation sequencing, and specifically in the realm of precision cancer therapeutics, which has been quite an exciting advent in our clinical laboratory medicine toolkit, if you will. So we'll start with some learning objectives for today. We'll talk specifically about the clinical utility of precision genomics, describe the advantages that clinical next-generation sequencing offers, as well as summarizing different general types of next-generation sequencing assays, and this is really in broad categories. And as we go through the presentation, that'll make a little bit more sense. So a recent New England Journal of Medicine editorial piece highlighted precision medicine and specifically called out three areas, saying that it's personalized, it's problematic, and it's promising, which I think summarizes quite well. I've added an additional bullet point to that, which is that it's also practical. So this is going to be our outline for today. So some physicians have really questioned whether we should be calling this personalized medicine or precision medicine, because they really feel that medicine has in fact been personalized for generations. And that's true in the sense that treatment has been tailored to a specific patient. Often when we're talking about precision medicine, we're talking about integrating not only biochemical markers, but really also integrating genetic and genomic markers to tailor a treatment to a specific patient. And we'll go through again some of these examples. I want to highlight that precision medicine does extend beyond genomics. So some examples are shown here. For example, in hyperlipidemia, when we're looking at LDL cholesterol, the treatment there would be statin therapy. And in HIV and AIDS, where we're looking at CD4 T cell counts to assess viral load, that would be then antiretroviral therapy. In pediatric cancer, where you have, for example, acute lymphoblastic leukemia, you would be using high-dose methotrexate in therapeutic drug monitoring, for example, to do leucovorin rescue. And then in lung cancer, if you have an EML4 ALK fusion, that would indicate that crizotinib therapy would be relevant for a patient. So again, this really does extend beyond just genetics and genomics, but that's been one advent that has highlighted the utility of precision medicine. When we're looking specifically at somatic genomics, precision therapeutics can really be guided by looking for specific genomic alterations. And so if you look at the patient population shown at the top here, so if you have cancer patients that are either metastatic or have recurrent or metastatic disease, or that have incident disease, there's a mixture of patients, those who have genomic alterations as well as those who do not have genomic alterations. So first, looking at the patients who don't have genomic alterations, you want to specifically identify those patients because they're not going to be responsive to a putative therapy. You want to identify them early so that you can put them on another treatment that will, in fact, potentially be effective for them. Not only are these drugs expensive, but they do, in fact, have side effects. And so we really want to maximize the therapeutic benefit. And then in patients where we can identify genomic alterations, you want to specifically match them to a targeted therapy. So it's important to recognize that it's not only that you have a mutation that's important in precision therapeutics, but it's just as important to not have an alteration and to figure that out early on in the process so that you can find the most effective treatment for that patient. And so the Human Genome Project used Sanger sequencing, and this is something that you may or may not have heard of as far as Sanger sequencing specifically is concerned. It's been really the standard of care in genetics and genomic research since 1975 when Frederick Sanger published the paper that discussed what exactly Sanger sequencing is. So Sanger sequencing, at best I'll say, can sequence up to 1,000 bases per reaction. So that's in a single continuous stretch, you can sequence up to 1,000 bases. Many graduate students, including myself, were really not consistently able to see 1,000 bases, maybe more in the range of 500 to 600 bases per reaction, but at best you can get 1,000 bases per reaction. It's a single molecule at a time that you're sequencing. And again, it has been widely used since 1975, and this was the technology that was used for the Human Genome Project. And so that project was an international consortium. It cost about $3 billion to sequence 3 billion base pairs of DNA. So it's quite expensive to sequence by Sanger sequencing, but that was really the standard. And there was some automated technology that was introduced during the course of the Human Genome Project that made that process be a little bit faster than originally anticipated, which is exciting. It's always good to complete a project more quickly than anticipated. But it still did take quite a number of years. And so one of the advantages that has come out of the Human Genome Project is that we now have this huge library of our genome, the human genome. And this is a picture that is from the Wellcome Institute, the Medicine Now collection, and I love, it's actually a printout of the human genome, and you can see there's a close-up here of one of these books. So it's per chromosome, you can see at the top. So if we look at 3.4 billion units of DNA code, that's how many nucleotides we have in our genome. So that would be 127 volumes if we have 1,000 pages per volume, and that print would be size 5 font. And you can't really see very well, but that A, T, C, and G, that's size 5 font. So it's quite a lot of DNA that we're talking about, and I use that really to illustrate the point that with next-generation sequencing, this presents a new challenge relative to what we were doing by Sanger sequencing. So with Sanger sequencing, you're directing your sequencing effort really to a specific question, whereas with next-generation sequencing, it's a little bit more challenging in order for us to pull out what you're specifically looking for. To talk very briefly about next-generation sequencing terminology, there's a lot of different terms that are used. So typically, first-generation sequencing is Sanger sequencing, whereas with next-generation sequencing, or NGS, as I'll abbreviate throughout the talk, that's also termed massively parallel sequencing or high-throughput sequencing. Relative to Sanger sequencing, then, it also becomes second-generation sequencing. And there are newer platforms available that are third-generation sequencing or single-molecule sequencing. And so, again, there's specific technologies that are used in each of these different categories. But for the purposes of our talk, all of these terms under the bullet next-generation sequencing, or NGS, can really be used essentially synonymously. So the National Human Genome Research Institute, NHGRI, publishes this figure, which you may have seen before, and it's a really good illustration. So I think that most people are familiar with the fact that sequencing costs have reduced over time, and that's what excuse me, that's what this figure is showing. And it's the green is specifically showing the cost per genome. This is per human genome, and the white line is Moore's law. And so this is really an amazing feat of technology in the field of computing. So Moore's law is the observation that the number of transistors in computing hardware has doubled approximately every 2 years. And that's essentially held true since the 1960s. So that's really been an amazing progress in the field of computing. And so this is almost a little tongue-in-cheek for next-generation sequencing relative to Moore's law. And you can see excuse me, that here is where we see an approximate inflection point. So sequencing costs were significantly decreased relative to there about 2007, 2008, and then we see this precipitous decline relative to that. And even we see again in 2015, there's another precipitous decline. And really what this is reflecting is the advent of next-generation sequencing. So it has had a significant effect in what we're able to produce per sequencing reaction, and it's really driving the cost of the genome down to the very close to the $1,000 point per genome. It's important to mention that's reagent cost, not necessarily interpretation cost. So if we at the broad level, very basically compare Sanger sequencing and next-generation sequencing, there's advantages that each method has. And so I've called out two specific platforms here just for the purposes of illustration. If we think about accuracy, Sanger sequencing wins. So it's about 10-fold higher per base with Sanger sequencing relative to next-generation sequencing. The throughput, we have a significant advantage in next-generation sequencing throughput. And then the amount of time per run, at this point, Sanger sequencing is still faster, even if you're doing 96 capillaries at a time, which is what we can do with some of these automated technologies. The read length is about fourfold higher with Sanger sequencing. So next-generation sequencing produces a lot of very short fragments relative to what you get with Sanger. The cost per meg base is significantly higher with Sanger sequencing, and again, this is what's really was reflected in the graph that we just were looking at. So it's about 8,000-fold higher by Sanger sequencing than next-generation sequencing. And there is a higher investment cost with next-generation sequencing. That's something that we will continue to see, I think, decline as we have more companies that are entering the marketplace. And if we think about what genetic assays really look like, they can range from genetic up to genomic. And so I've shown this with, going from single site, that would be with a single base pair here, all the way up to genomic, which is if we were looking at the entire human genome. And perhaps a little bit more fun to think about it is relative to ice cream cones, if you will. So a single site is if you're looking, again, just at a single base pair change, and then looking at a continuum from a partial gene somewhere in the 100 base pair range up to different types of panels. An exome is the coding sequence, so the protein coding sequence of our genome. And then, of course, the genome would be the entire human genome. And I've called out some specific examples here. So a single site relative to a cancer change would be like if you have a BRAF codon 600 mutation. So often in the case of melanoma, you would be looking at that specific mutational profile. Thinking about partial gene or genes, for example, in cystic fibrosis screening, where we're, for carrier screening, typically looking at 23 mutations. And then, again, there's hotspot panels, comprehensive panels, and then extending up to exome and genome. So it's typically not 100%, but often when you get to something that's called a hotspot panel, that next-generation sequencing really has both utility and cost-effectiveness. So you need to be looking at more than just a few base pairs or just a partial gene. Again, not 100%, but often before next-generation sequencing starts to become cost-effective. And we'll look at some more examples of what that specifically is, again, as we go a little bit more in depth. So going back to our outline, we've talked a little bit about the personalization, but let's talk about the practicality. So next-generation sequencing is clinically offered in diagnostic laboratories around the world today. So it is, in fact, already in the clinic. And there's a lot of utility in thinking about assays from a multi-gene perspective. So historically, we've done really single-gene testing. And so when we're doing that, we're looking at a limited region. So you want to look specifically at a single base pair change. By definition, then it's narrowly targeted. And that result may trigger additional gene testing. So based on whether you get a positive or negative in your first gene, you may go on to do a second, third, or fourth gene. If you compare it and contrast that with multiple gene testing, you're looking at all relevant genes. And again, to focus on cancer, we would be looking at not necessarily all relevant cancer genes for a particular cancer diagnosis, but really doing potentially a pan-cancer assay. And that's what's shown here, where you would be looking at BRAF, EGFR, KRAS, FLT3, KIT, and NPM1. So simultaneously, you detect the mutation status of all of these genes. And that's really a paradigm shift from how we've traditionally done this. It is cost effective. It's efficient in that you save time. And really important is that it's tissue sparing, particularly as we move in the field of oncology towards smaller and smaller biopsies. And you've probably heard liquid biopsies, but even in the solid tumor realm, to think about fine needle aspirates and other very small pieces of tissue that we acquire. We really don't have enough tissue to go from one assay to the next to the next. So if we can have one assay that incorporates really all of the relevant mutations for that cancer type, that's very powerful. And then this type of testing can certainly yield unexpected findings. And we'll look at some examples of that. I have some case examples that we'll discuss. And it's important to think about that both in the realm that it can produce unexpected findings that you expect, so mutations in other cancer genes, as well as mutations that you don't necessarily expect. You can find alterations that maybe are actually associated with the germline. And again, we'll talk a little bit more about that. So there's a recent review paper that highlights some of the different technologies that are used for next-generation sequencing. And so there's a lot that's actually on this slide, so we'll talk a little bit in depth about it. The time is what's shown across the x-axis from 2004 to now, and then on the y-axis we see the machine output, and this is sequenced in megabases. Okay, and then for reference, the 100x exome versus a 30x genome are shown as well. And so there's there's a number of different companies, and that's what's shown in by the color of each circle. It's a little hard to see, but there are, inside the circle, there's a number. What that number is reflecting is the base pairs per next-generation sequencing read that you get. So you can see that they range from very small. So for example, here we have 35 base pairs up to very large. So with the Oxford Nanopore, we get 6,000 base pairs potentially per sequencing read. But most next-generation sequencing platforms really give you a very small, so in the 1 to 200 to 300 possibly base pairs per sequencing read. And there are two platforms that are predominant in the clinical laboratories, and those are the Illumina platform and the Ion Torrent, which is actually now owned by Thermo Fisher. And so those are what are the dots that are in red and the dots that are in blue here, just for reference again, because those are predominant in the clinical laboratories today. The general workflow for next-generation sequencing varies from one platform to another, but particularly when we're thinking about those technologies that are dominant in clinical laboratories, it starts with template preparation. That can be either DNA or RNA, which is an important point to emphasize that you can, in fact, start with RNA if you're doing RNA-seq. From there, you do massively parallel sequencing, which both the ion torrent and the Illumina use clonal amplification, and that's followed by some form of detection, whether that's imaging or detecting a pH change. And then data analysis follows that, which involves genome alignment and assembly, and of course more sophisticated analysis depending on what the specific application is. The read alignments are really the raw data if you think about next generation sequencing. So we'll spend a minute here to look at this. Down at the bottom is the reference sequence. And so this is showing FLT3, which is an important gene involved in cancer, specifically in AML. So it has prognostic significance. So the reference genome are these tiny little lines that just show up as different colors. So when we do next generation sequencing, we align back to the reference genome, and what we get are a number of different individual sequencing reads you'll hear. And so that's what's shown in all of these little blue bars are the sequencing reads. And then at the top is the aggregate coverage, and this is what is shown in the gray. So that is the coverage that we're getting. And you can see that the more blue bars that we have for an area, the higher coverage we have. So this is often called the depth of coverage when people are talking about sequencing. So it's really, it's relative to how many sequencing reads we have at a particular locus. And so what we're talking about is, that you would have many cancer assays tend to have in the range of 500 reads per sequence base. So about over the entire assay range, you have on average 500 reads per sequence locus. And so in cancer diagnostics or even in constitutional changes, there's a number of different types of variants that exist. So they range from a single base substitution, and again, I'm using BRAF as an example where you have a V600E change. Again, that's very prevalent in melanoma. You can have small indels. Indel stands for insertion deletion. We just abbreviate that with indel. So in lung cancer, for example, we see exon 19 deletions in EGFR. And then you can have medium-sized indels that are typically referred to as less than 500 base pairs. Large indels are those that are greater than 500 base pairs. And an example of that is if you specifically have an exon deletion, for example, in TP53 that's associated with Li-Fraumeni syndrome. That's a cancer predisposition syndrome. Copy number variants are typically referred to as those changes that are greater than 1,000 base pairs. And so, for example, we have MET amplification that occurs in multiple different cancer types. And then structural variants are highly variable in size. For example, a translocation. And one example of that would be EML4-ALK, which is a recurrent mutation that occurs in lung cancer cases. So it's important to recognize that variants do in fact come in all shapes and sizes, because the way that we detect these when we're doing our next generation sequencing assays is different. So for basically each type of change, you need to have a different bioinformatics solution in order to pull it out. The powerful piece of this is that one assay can actually determine all of these things, but it's really in how you analyze the data determines whether you can pull out individual aspects of each of these changes. So currently, there are a number of clinical applications of next-generation sequencing that are commercially available. And so those range from somatic, specifically in cancer, of course, to inherited disorders, and that's really a wide array of things. So cancer predisposition, there are a number of assays for different types of mutations that are grouped by pathogenesis, or even exome, for example, if you have a seemingly Mendelian disorder, but you can't identify the mutation that's underlying that. There's also non-invasive prenatal testing, and so that's using next-generation sequencing by most commercial assays. And then there's even more exciting microbial identification, and this is where we're starting to do next-generation sequencing to specifically pull out bacterial, viral, and other pathogens, specifically looking for their genome. And the computational side of that is quite challenging, but it's very exciting that this is something that's now commercially available. So when you're thinking about an assay design, whether you would be looking on a genome level or doing smaller scale really depends on the clinical need. So for non-invasive prenatal testing or microbial identification, whole genome sequencing is really the best choice for that because you need low coverage, so you don't need to sequence each base a lot of times, but you really need a sense across the genome. So for example, in non-invasive prenatal testing, it's important that you're picking up all of chromosome 21 at low coverage, but you really want to see all of chromosome 21 to assess whether you have trisomy 21, for example, for Down syndrome. For inherited disorders, often that's exome sequencing. So again, it's fairly low coverage, but it's across all of the coding sequence of the human genome. And then if we're thinking about specifically for somatic testing, this really tends to be what's called targeted sequencing. And the coverage is much higher. That's important because in somatic sequencing, we need to be able to detect both the germline and the cancer, so those coexist in a single specimen. So we get a biopsy specimen that has not 100% cancer cells. So we have a mixture of cells. And that targeted sequencing can be either comprehensive, so determining really all of the relevant cancer genes at once, or it could be looking at hotspots. And so, for example, that we're only pulling out the mutations that we know are very important, either in a single cancer type or in multiple cancer types. So I have a single case example here that we'll look at over a couple different slides. So this is a patient with advanced lung adenocarcinoma. He's a 71-year-old male with metastatic lung adenocarcinoma, and that was diagnosed in February of 2014. And so he had a poor response to first-line chemotherapeutics. And if you look at the NCCN guidelines, what they recommend is to do molecular profiling for this patient in this context in order to guide treatment, right? So he's already received the standard of care, didn't have a good response to that. And in order to choose a subsequent therapy, they recommend doing molecular profiling to really help assess what treatment would be most helpful for that patient. And part of the reason that they recommend doing molecular profiling is it's shown in this slide. So recurrent mutations that you see in lung adenocarcinoma are really quite extensive. So they range from, KRAS is one of the most common things that mutations that you'll see, and that actually indicates that you would not respond to typical tyrosine kinase inhibitor treatments. But there's a number of other mutations as well. So EGFR also has a fairly large piece of the puzzle, or piece of the pie here, but there's a number of additional alterations that are very low frequency events, but nonetheless are recurrently mutated and often have associated therapies. So that's one of the reasons that, again, molecular profiling is particularly prevalent in lung adenocarcinoma, and that's what the NCCN recommends. So we'll come back to this case vignette after we look at a little bit more depth what type of assays there are available. So in profiling assays, there's very broad division. We typically refer to them as hotspot assays and comprehensive assays. So I've listed some specific examples and then just a pictorial representation of what a hotspot assay would be. So in this case where, again, you're specifically pulling out particular mutations that you know are recurrent and important in a cancer type. So what that gives you is maybe an incomplete picture, but you can tell from this picture that this is a boat, right? You can't necessarily tell, for example, that there's a flag hanging off of the back of the boat from the first picture, but you could get a general sense. With a comprehensive assay, and again, I've shown some examples here, what you get is a more complete view of the cancer type because it depends on how you've designed the assay, but typically what you're trying to do is really to identify all mutations in the relevant cancer genes, because those can, in fact, have important significance for selecting the specific therapy for a patient. So just at a very broad level, to compare hotspot sequencing and comprehensive sequencing, in turnaround time, hotspot sequencing is definitely faster. That partially reflects how the DNA is prepared even before you do the sequencing. But even after you finish the sequencing, the downstream analysis side of that is definitely faster. At this point, you do need less sample when you're doing hotspot sequencing than relative to comprehensive sequencing, but there have been some important advances that we've seen in the library preparation. So this will continue to, I think, be something that we'll see improve over time. Single nucleotide changes can really accurately and adequately be assessed with either method. I put a plus minus for hotspot sequencing just in that you have to specifically design your assay to detect whatever mutation you're thinking about, whereas with comprehensive sequencing, again, you would already detect these. And so for small indels, that's typically something that you can see with hotspot sequencing. But then when we get into copy number changes or structural variant changes like gene fusions, that's something where comprehensive sequencing has a significant advantage. The informatic analysis is significantly easier if we're doing hotspot sequencing than in comprehensive sequencing, but the ability to identify novel variants is definitely easier with comprehensive sequencing, and then cost is lower with hotspot sequencing, as you may expect. So to go back to our case example with these two broad types of sequencing assays, so this patient has advanced lung adenocarcinoma, and so there's all these different types of mutations. At this point, the hotspot assays aren't able to detect the rearrangements that are shown in red boxes. So in this context, it's probably one of the most cost-effective ways to start to look for changes for this patient that would, again, change his specific treatment plan, would be to do a comprehensive sequencing assay. So let's talk about some of the challenges and limitations that arise from doing clinical next-generation sequencing. One important question, I think, is to ask, is more always better? So when we're thinking about large sequencing panels, we can do more, but does that actually have a clinical benefit? So some of the challenges are the interpretation is significantly more challenging in order to be able to detect these multiple types of changes. That takes longer both for the bioinformatics as well as for the analysis. The turnaround time is definitely longer. At least in the U.S., the reimbursement is definitely something that's still evolving in this field, so there's not universal acceptance by different payers of the CPT codes that are used. And then you can have secondary or incidental findings, and that's when you find something that you weren't specifically looking for in your assay. And there's a lot of different types of things that can be found. But the advantages include that this is, in fact, comprehensive, which leads to at least a higher hit rate for diagnostic and or actionable mutations. And that's important for driving both clinical trials and discovery. And typically, it depends on the specific test that we're talking about, but they're typically priced such that it's cost-effective if you're sequencing more than 3 genes relative to doing it individually. So this large panel, for example, a panel that we do sequences more than 250 genes. So if you were going to do individually 3 or more genes, in this context, getting more than 250 is actually significantly cost-effective. It's cost the same to get our panel as to get 3 individual genes. But these assays are laboratory-developed tests, and as such, they really have to be rigorously validated. And there's still not as much guidance as there otherwise could be. So really, the early adopters, some of the professional societies and regulatory agencies do provide guidance, but it's not as strong, we'll say, as some of the guidance that you get in other areas of the clinical laboratory. And it's definitely an area that's continuing to evolve. relative to the FDA, for example, for laboratory-developed tests. And it's important to recognize that a number of different types of variants are poorly or not detected by current next-generation sequencing, either the platform or the pipeline, and it's really both together that you have to consider when thinking about the clinical need. So substitutions in small indels are not that challenging for us to detect by next-generation sequencing, and that's really irrespective of the platform. But once we start to get into larger insertions and deletions, copy number variants, and structural variants, like translocations, that's definitely much more challenging, and some platforms aren't able to detect this at all. So that's, again, something that has to be really carefully validated in a clinical context. And I put up here the central dogma of molecular biology going from DNA to RNA to protein to really emphasize that what we're looking at if we're doing next-generation sequencing from DNA is we will see a DNA change. If you have a change that's downstream of DNA, that of course changes whether you're expressing a protein, that's not something that we would be able to detect with current methods. And there's a number of bioinformatic challenges that arise from next-generation sequencing, and what's shown here is really some of the important interpretation piece of this. So this is an example of a TP53 frame shift deletion, and that's what this black bar is showing you a deletion in TP53. And so in this case, we had 500 reads at this particular base, and 56 of the 500 showed this deletion. TP53 is an important cancer gene, so you may think that this is, again, an important change that we're seeing that's really contributing to the patient's cancer type. But we have in our laboratory an internal assay-specific frequency database, and so across all of our assays that we've done, we see this mutation in 82% of our cases, right? And so what that's telling us is that this is an analytical artifact. It's not contributing to the patient's cancer because we see, again, this is in 82% of the cases. And so it's really important that you have this built into your pipeline to be able to detect these recurrent artifacts because the error profile of next-generation sequencing is significantly higher than what we see with some other technologies like Sanger sequencing. And one way that we're able to get this sensitivity that's really important for the clinical context is through parallelization. And so I'm using that term to emphasize that different bioinformatic tools, for example, different variant callers, really have different strengths. And so stacking multiple approaches into a pipeline can significantly improve your sensitivity. So in this example, we have the variant caller 1 over here that detects a certain number of mutations, and then the variant caller over here that also detects a certain number of mutations. But you can see that it's not an overlapping Venn diagram, right? So we have some that are only called by variant caller 1, and then we have others that are only called by variant caller 2. And what that leads to really are different strengths and limitations of each. So we have some variant callers that have a high degree of sensitivity. Excuse me. And so what that high sensitivity means is that we have a number of true positives, but we also get cases like here where we have a false positive, whereas, for example, by variant caller 2, we have a caller that's high specificity, so we're not necessarily detecting all the changes that are there, but we have almost all the changes that we identify are actually true positives. And so it's really the intersection of these two that can provide significant benefit on a clinical assay. The way that we approach this in our laboratory is to stack both variant caller 1 and variant caller 2, return all the data to the director, and then really use our clinical expertise in filtering that list down and assessing whether a change is clinically significant or not, or some sort of an analytical artifact. And that's how we avoid reporting false positives. But it is definitely a manual process. So the clinical reporting model that we use at the University of Washington is quite integrated. So we start with a multidisciplinary team that contributes to the panel design. And so then we have, once that's implemented, the data analysis and mutation identification is something that occurs in the laboratory. And then we have a consensus team that really interprets each case. And so that includes laboratory directors, clinicians, and genetic counselors, laboratory genetic counselors. The report is specifically written by the lab director, entered into the medical record. And then this last piece is particularly important, where we assess the treatment and response, and then we feed that back. So it's a constantly evolving process of knowing what the changes that are identified in a clinical sequencing case really mean in the clinical context for a specific patient. So again, it's an iterative process. And so incidental or secondary findings are really a hot button issue, if you will. So the American College of Medical Genetics and Genomics had issued recommendations around this topic in 2013, and really recommended in this initial publication that all clinical exome and genome sequencing should report back from this minimum list of 56 genes in both adults and children. So if you identified mutations in these genes, they really needed to be excuse me, reported back. And they were specifically advocating a proactive search, if you will. So if you were sequencing, let's say, the cancer predisposition gene BRCA1, even if you weren't pulling out mutations, but if your assay was able to sequence that gene, that you proactively went in, searched for whether there's mutations there or not, and reported that back to patients. And this was extremely contentious, so you can see in a fairly short period of time, that article had a number of citations. And ACMG did update that recommendation in 2014 to allow patients and physicians to opt out. But it's definitely a field that's continuing to evolve, and as I mentioned, contentious at best. So let's talk about some specific promising examples that we've seen arise out of clinical next-generation sequencing. So if we focus on lung cancer, and that's, remember, the clinical vignette that we've used previously. So this is one comprehensive cancer assay. There are others that have shown similar findings. But across over 500 cases, what we found was that there was an actionable variant that was identified in almost half of the cases. And it was similar across all cancer types, but we've called out lung cancer just because we had a lot of cases in this initial cohort. It's really important to emphasize that when you're thinking about the yield of a test, it really depends on what your definition of actionability is. So some publications will use the definition of actionability as that you just find any recurrent mutation. So if you find a TP53 mutation, for example, in lung cancer, that's not actionable. You're not going to treat a patient differently based on that mutation with a definition that was used in this particular publication, but other publications do use that. So it's just something important to keep in mind when you're thinking about what, quote-unquote, actionability really does mean. KRAS, it does tell you, for example, not to use a specific treatment. That's how it's being used in this publication. And importantly, we can identify mutations, but this was really a seminal study in saying we can identify mutations, but it actually has a significant clinical impact. So what we see, and this is again in non-small cell lung cancer, so if patients if they had sequencing, so if they had clinical sequencing, that's what's shown in either the darker blue or green here. So they had sequencing, but there was no targeted therapy. But over here, if they did have targeted therapy, versus those where if no driver was actually identified. So what you see is, if you do sequencing and there's a matched therapy that a patient is able to be put on, they had a significant advantage as far as overall survivability. So again, that this does in fact have clinical benefit to do molecular profiling in this patient population. So going back to this case vignette that we we've used a few times. Remember, this is a 71-year-old male who has metastatic lung adenocarcinoma that was diagnosed in February of 2014. He had a poor response to the standard of care first-line chemotherapeutics. He had a comprehensive next-generation sequencing test that was performed in March of 2014. And this is the mutational profile that we know exists in lung adenocarcinoma. That's why, in this context, they selected a comprehensive next-generation sequencing assay. And so what we found in this patient was a mutation in the MET gene, and the specifics is shown here. And the effect of this mutation is that the protein half-life is increased, so that leads to higher levels of MET activity. Excuse me. And so when that signaling pathway is overactive, what we know is that crizotinib is an effective treatment to use. And so that was used in this patient, and he tolerated it well. And 47 days after therapy was initiated, he had a follow-up CT, and the radiology report specifically said there was a substantial interval decrease in the lung metastases, and he had a continued response at 90 days post-initiation by CT. He's at this point being followed by his community oncologist, so I don't have follow-up CTs after that, but that was, definitely a benefit that this patient received and really wouldn't have gotten without doing a comprehensive cancer sequencing assay. So looking at another context is in clinical trials. So in this context, we have a 58-year-old female. She has recurrent metastatic breast cancer, and it's both estrogen receptor and progesterone receptor positive. She was diagnosed and treated in 2000, and then they ordered tumor sequencing for treatment after she had recurrence over 10 years after her initial treatment. And what we saw was that there was FGFR1 amplification that was present, and this is showing us specifically how we look at our copy number variants. And so on this axis we have the chromosome, and then the number of gene copies are shown over here. And then I've specifically boxed out FGFR1 because we do see amplification of that gene. So she was enrolled in an FGFR1 inhibitor clinical trial. And again, this is one of the advantages of doing comprehensive next-generation sequencing. We can detect really all of the mutations that are important, and that can really further clinical trials in discovery and development of new treatments. And so it's really patients who are treatment refractory who may have the most benefit from doing comprehensive sequencing. So this is a 56-year-old female who was a never smoker who presented to her primary care provider with a neck mass, and they identified poorly differentiated lung adenocarcinoma that was unfortunately metastatic, already at stage 4 at time of presentation. And that's, again, unfortunately not an uncommon occurrence that we see in lung cancer. So often these patients are presenting later in their course of disease. And so they ordered tumor sequencing for treatment because she was so advanced at the time. And what we identified was a ROS1 gene fusion. And if you have to have lung cancer, I guess I would have to say that this is the best kind that if I could choose, this is the kind I would choose. So ROS1 has a specific targeted therapy that you can use in this context. The ROS1 inhibitor was initiated right away. It was well tolerated, and so 2.5 years later, the patient is stable. There's no signs or symptoms of her lung adenocarcinoma, and she works full-time, owns her own business, and her new goal is to be certified as a spin instructor. So those are the type of success stories that we definitely love to see. And then as a final example, going back to this example I mentioned earlier, in that if you have a change in your DNA, we can detect that quite easily with next-generation sequencing, depending on which type of change it is. But if you have a change in your RNA, if we're sequencing DNA, we won't detect that. And so what that change can lead to, of course, is an aberrant protein. And so this is another success story from the group at Washington University in St. Louis, and it was published in the New York Times. And this was a case where there was a change in RNA, and it was a specific gene that was mutated where there was a specific inhibitor for the gene that was mutated. So sequencing the DNA, they weren't able to identify the change, but they used RNA sequencing to see that they had significant overexpression of that messenger RNA, which was leading to then significantly increased protein concentrations. And using that specific inhibitor, they were able to effectively treat this patient. And if you want to read more about that, it was published in the New York Times. So just to conclude, so there's been a lot of editorials talking about genomics, whether it's hype or hope. And so this was something that Francis Collins had published at the time that the Human Genome Project was completed. So it's an editorial from 2006. And so I really like this figure where the enthusiasm cycle for a new technology is what's shown. And so if we look at over time on the x-axis, we compare that with the level of excitement on the y-axis. And so you have the initial idea followed by enthusiasm, and so you have this this peak where you have a lot of hype, right, followed very quickly by a reality check. And so you see the period of disillusionment following, and then you gradually increase to really having the true performance of an assay. And I think that this is really where we're at now with clinical next-generation sequencing, where we're starting to see the technological advances meeting the clinical needs, and that will only continue to increase as we see additional products come to market that it really do have demonstrated clinical utility and clinical benefit. And so I'll conclude here with some acknowledgments, groups that I've collaborated with both at the University of Washington as well as Washington University in St. Louis. And I would be happy to take any questions that may be coming in through the Q&A.
Thank you very much, Dr. Lockwood, for bringing that informative presentation to us. Before we get started with our question and answer session, I want to remind the audience how they can submit questions for you. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower level of the presentation window. And hopefully we'll have some good questions, and we'll try to get to them as many of them as we can. That was definitely an interesting presentation, Dr. Lockwood. I hadn't heard anything about this type of sequencing. And we do have our first question. Marina from Morphogenesis asks, are there clinically available panels that screen how tumor cells escape the immune system in addition to common tumor mutations? For example, CTLA4, PD-L1.
It's a very interesting question, and so thank you for bringing it up. So these types of treatments, so for example, anti-PD-L1 are really becoming more. These immunotherapies are becoming more prevalent in cancer diagnostics, and it's definitely an exciting area that will have a significant benefit. We're starting to see this already in melanoma in particular. At this point, there aren't. There's certainly not any sort of a commercially available package that's really able to identify this. It's happening in the research context, and so clinical laboratories, similar to how we've developed these, our comprehensive tumor sequencing assays, clinical laboratories are really developing their own tools to look at this. So it's something we've seen come out of the clinical trials of the specific immunotherapies. But there is not a commercial product that can be purchased. It's something that would have to be developed de novo.
Thank you for that answer. It was a good question from our audience member about this kind of technology. I'm just going to give our audience a minute to submit any more questions that they have, and we'll watch the question queue for that. If we don't have any further questions, I'd like to ask Dr. Lockwood, do you have any final comments for the audience about this topic and about where it's going in the future?
I'll just emphasize, you've heard throughout my talk several times I've talked about how exciting this technology is. I definitely think that we're going to continue to see more growth in this area. both as far as more laboratories offering it as well, and commercial products that are coming to market. So I think that's something that we'll watch and be excited to see continue to develop. And as our audience member has already asked, it's not only going to be in the we identify a mutation and that identifies a treatment, but I think we'll see expansion beyond that as immunotherapy comes more to market. And it's really going to be the integration of multiple technologies that are going to lead us to this, I think now hope, not hype, but hope of precision cancer therapeutics.
Excellent. Hope, not hype. That sounds fascinating. I want to let our audience know, now that we don't have any more questions, that today's webcast will be available for on-demand viewing through April 1st. Any of our audience members who are here today will receive an email from LabRoots alerting them when the webcast is available for replay. And we would invite you to share that email among your colleagues who perhaps could not make today's live event. I want to thank Dr. Christina Lockwood again for bringing this information to us. We hope to see you next time. Goodbye.
Thank you.